First of all, I'd like to thank Chris for the invitation to come here and share with you some of the work that we did back in 2008 to 2007, and then thank you all for being here today. I'm a research entomologist with USDA, ARS, and our year. We have one project with 8 scientists. All the 8 scientists work on Xylella fastidiosa. It's a bacterium. I'm the lead scientist on the project. This bacterium here invades the xylem vessels of plants. They invade the vessels, they start multiplying, they clog the vessels. Water doesn't move very well towards the top. And then we get scorching symptoms, we get dieback on a number of different plants. It's native to the Americas. It causes disease in plants in South America, Central America, North America, up to a certain latitude. It's been found in Taiwan, probably transported in plant material from California to Taiwan. It has been recently found in Europe, affecting some plants, or not. That's what we're trying to find out. This is a bacterium. It's not new to California. It's been around for a number of years. Back in the 1800s, the late 1800s, there was a disease in Anaheim. It was called Anaheim vine disease. It took about 60 years to find out it was caused by Xylella fastidiosa. It was thought to be caused by a virus. And you can see here the symptoms of scorching. We get the scorching on the leaves. We get some green islands on the bark. Disease was finally described. As I mentioned, it's been causing disease in a number of plants, different parts of the Americas. They're divided into subspecies groups. That's very important here. That's based on their genotype. What people do, they sequence 8 to 10 housekeeping genes. Those are genes very important for the bacteria, very conserved in the species. So they get the sequence of that DNA and they can separate them into subspecies. So in California, we have Xylella fastidiosa, subspecies fastidiosa, causing fierce disease in grapes. We have Xylella fastidiosa, subspecies multiplex, causing almond leaf scorch disease. Fastidiosa will also cause almond leaf scorch disease. Phony peach disease in the eastern part of the United States, affecting peach. Plum leaf scald. Citrus variegated chlorosis in Brazil, Argentina, parts of Uruguay, and that is caused by Xylella fastidiosa pauca. That's a different subspecies. That subspecies is not known to occur here in the United States. We're not aware of any infections like this particular subspecies here. Everything we have, it's either fastidiosa or multiplex group. In Taiwan, it also calls pear leaf scorch. That appears to be a different species. We don't know yet. People are doing whole genome sequence analysis nowadays instead of just sequencing genes. Apparently, there's a pear leaf scorch disease in Oregon, which was reported 2 or 3 months ago. It has not been confirmed. There's actually Xylella causing the disease, although Xylella has been found on those trees, but people need to do some experiments to demonstrate that it's actually causing disease. And there are a number of other plants that are affected by this bacterium, ornamental trees. It's a xylem limited bacterium, so it's transmitted by sharpshooters that feed on xylem, much like mosquito-borne disease. The mosquito will feed on an infected person, transfer the pathogen to a healthy individual. So these guys will insert their stylets in the plant. You can see glasswing sharpshooter here, inserting the stylet and the xylem, just ingest xylem for a number of hours. Bacteria will attach to their mouth parts, they'll fly to a new plant, then the bacteria will get inoculated into the xylem of a new plant. In California, there are about 14 species of sharpshooters that can transmit Xylella fastidiosa, but everywhere you have Xylella, you have a different vector. If you go to South America, they have a huge complex of vectors. If you go to Taiwan, they have a different vector. If you go to Italy, they have a different vector. So these are the 4 that are more important in California. And they're kind of divided in areas. The blue-green sharpshooter is more common in riparian areas, in coastal areas, coastal vineyards. The green sharpshooter, they are common here in the Central Valley, mainly on irrigated pastures and areas that have grass available all year long. If you mow the grass for 2 months in a year, you can knock down the population. Glasswing sharpshooter is very invasive, so it's in Southern California. It's been found here in it actually has established population here in the southern portion of the valley, and I'll talk a little bit more about that in a minute. So we're talking about a vector-borne disease, a vector-borne pathogen. There are many ways that they can spread the bacteria. One of them is primary spread, so we have individuals flying in from outside areas into the orchards, inoculating these plants. And then you also have secondary spread. When you have established populations of vectors in the orchard, they start moving the bacterium around. So these are two very important aspects that I'd like you to keep in mind for a while. And this is an example here in Napa Valley. It's a classic photo. This is a vineyard. So the blue-green sharpshooter, during the winter, they'll stay in these riparian areas where they will acquire the bacterium from different plants. Bacterium can infect more than 300 plant species. So they can pick up the bacterium here and then move into the vineyard. So you have this pattern of primary spread. There's not much secondary spread because pruning helps a lot at the end of the year. People come and prune. That way they avoid a systemic plant infection. There's no time for the bacteria to actually move down to the roots and become fully systemic from the plant. So pruning helps a lot. So primary spread in the northern parts of the state here. Then glasswing sharpshooter came in, invasive species. It's native to this part here, North America, northeastern Mexico, and it made its way to California. They believe that it was transported in ornamental plants. And it liked California very much. It was first found here in Irvine. There was a rapid population growth and range expansion. A few years later, it was found in the southern portion of the San Joaquin Valley. There's been a number of findings here up north. These are data from CDFA 2008. I don't even bother updating this slide anymore because they keep finding glasswing sharpshooter in shipments of ornamental plants from Southern California. Just last year, I think they found them in the olive plants transported from Ventura County to Marin County. But as soon as they find it, they put some effort to eradicate that population there. What they don't want is this insect to establish in Napa, Sonoma area. With glasswing sharpshooter, we don't see the pattern that I show you with blue-green sharpshooter, that primary spread invading the vineyard. We think because these insects, they fight deeper into the vineyard. So there's no pattern. Pretty much, this was in Temecula when sharpshooter was first detected. It destroyed 130, 150 hectares of vineyards there. So pretty much everything was gone until they start treating for the vector, reduce the population, and now vines are growing back in Temecula due to the area-wide management program for glasswing sharpshooter. So that's sort of an introduction to Xylella and their vectors and what we have in California. I guess the reason why I'm here today is to talk about olive leaf scorch disease, but I'm going to focus on Xylella fastidiosa. And that's the work that we did. We got together with the California Olive Committee a few years ago. There was an olive leaf scorch disease, dieback disease in California, but people didn't know what was causing the disease. So I assembled my team. These are my colleagues in Parlier. Mark's also an entomologist, JC a bacteriologist, Drake is a virologist, Marsh Johnson with the UC Cooperative Extension with funding provided by the California Olive Committee. And so the problem was these increasing occurrences of olive leaf scorch in Southern California that eventually will lead to death of the plant. Not only in the LA area, but also in Santa Barbara. So people became concerned. We got together with the California Olive Committee, a group of researchers from UC Davis, led by Doug Gubler. They looked into fungal pathogens, and they did find some about 18 species of fungi infecting olives. At least 3 of them caused dieback in olives. But we don't know much about olive leaf scorch disease right now. And what it looked like, this is a picture from Southern California. It looked like someone just went in with a torch and started burning these trees. So we go to Southern California, and what I found quite a few olive orchards. They do change the spacing between trees and rows so they can park their cars underneath, but they're basically they're olive orchards. These are trees that were transplanted into these parking lots. So it's not really a commercial orchard. On those trees we find glass-winged sharpshooter. Here's a young male on the top, no female here, the nymphs on the stem. And they're not hard to find, especially on these suckers that come out of the bottom of the plant. These are all nymphs. They prefer to feed on those because it's more of a tender tissue, easier for them to reach the xylem. And you can find egg masses, you can find nymphs, and you can find adults all at the same time on the same plant. There's overlapping between generations there. So they do like olives. They use olives to overwinter. It's an evergreen plant. The other most common host for glass-winged sharpshooter in California is citrus. It's actually the most common, and perhaps olives as well. You see a lot of scorching, so I had to talk about olive scorch. So here's my slide on olive leaf scorch. There are a number of different scorching or scorch types. They're kind of an inverted V where you have this very sharp transition from brown to a green with the yellow. In between, some are not that regular, they're more irregular, scorching on the tips. These are kind of more long, drying of the tips of the plants. We also see dieback, some extensive dieback in these urban environments. Some trees are completely dead. Young trees and also older trees. You can see one planted on a nice green lawn here. And that's me collecting samples in La Mirada. A tree that was positive for Xylella fastidiosa, and it was under tetracycline applications, just being injected directly into the trunk. So different types of leaf scorching symptoms, and then dieback. Our research questions were: how prevalent is Xylella fastidiosa in California? How widespread is Xylella in olives in California? Which subspecies are infecting olives in California? Which vectors are involved in the spread? Does it really cause disease in olives? And olive strains of Xylella fastidiosa can cause disease in other plants, for example, grapevines and almonds. So samples were collected, photographed. Every single sample was brought to the lab, labeled. So you can see here different kinds of symptoms, dieback and leaf scorch. All these samples and many more tested positive for Xylella fastidiosa. That doesn't help when you're trying to describe a disease because you're looking for an association between a symptom and a pathogen. If you have a symptom and you always find that pathogen, there's a constant association, then that indicates that that pathogen, that one organism is possibly the causal agent of the disease. In this case, all sorts of symptoms, they're all coming back positive for Xylella fastidiosa. This is just a summary of our sampling. Most of the sampling was done in District 3, so 4 samples collected in Davis. So I did most of my sampling in Southern California. About 50% were done in the San Joaquin Valley, 50% of the samples from Southern California. These are the numbers that were positive for Xylella fastidiosa by PCR, like the pregnant test that Jim described. Grind the tissue, it pops up a positive sample, it is infected by the bacterium. In the San Joaquin Valley, we found only 3 samples that were positive for Xylella. That was in Ducor. That's an area that had in the past had a heavy infestation by glass-winged sharpshooters. Then the area-wide program kicked in, populations dropped. But I think that's just a residue of glass-winged sharpshooters moving this bacteria around. And the vast majority of the samples came from Southern California, where these insects are just widespread and doing very well. Of these plants, we got 6 isolates, so that's something that we grow in the culture and we can actually characterize. Before characterizing, we used JC, my colleagues, he developed a 4-primer PCR technique that basically tells us if one particular isolate will cause Pierce disease in grapevines or it will cause almond leaf scorch disease or both. So we did the preliminary 4-primer PCR, and that showed that these strains, these isolates, would cause almond leaf scorch disease, not Pierce disease. We follow up with a multi-locus sequence typing analysis. That's the one that I mentioned, that we sequenced the housekeeping genes of these organisms here. And what we got, here we have the 985C, that's a subspecies Falca from South America that causes citrus variegated chlorosis. We used it here as the outgroup, so we can do this analysis here. The fastidiosa clade or group is here. That's the one that causes Pierce disease in grapevines. And here's the multiplex group that causes almond leaf scorch disease. Our olive isolates, they grouped with M12. They were 100% similar to M12, which is a strain that causes almond leaf scorch disease. So that suggests that the strains that we got from olives would cause almond leaf scorch disease and not Pierce disease. We still need to follow up with inoculation experiments. We can't just go out and say that because they grouped in here, they will or will not cause disease. We're not at that point yet with this kind of analysis here. So what we did, and that's called Cox postulates. Basically what it is, we have a disease. It can be biotic or abiotic. That would work for abiotic disease. But there is sort of a modification of that for confirming biotic disease. So we have a disease, we isolate the pathogen, purify, we do transfers to make sure that we're actually working with the pure colony of the organism, put it back on the healthy plant. Then we need to reproduce the symptoms that we see initially in the field, and then re-isolate the organism from that plant. And I'll tell you right now that what we did was this part. When we got to the symptoms, it was kind of a messy situation there. I'll come back to that in a minute. The methods we used for this experiment was meat inoculation. So we prepared inoculum and just inoculated these bacterial cells in the plant. 284 plants, 7 different varieties. Here they are. And then we did a detection of the bacteria later on at 3, 6, and 12 months after inoculation by PCR and also culture, just trying to grow it back from those plants. For the results, we had, of the 284 plants, I think there were about 15 or 18 that showed symptoms of leaf scorching, not much like the symptoms that the isolate came from. And then some had a dieback. Here are the numbers. So 18 of the 284 plants showed symptoms of a dieback or leaf scorch. However, we saw the same symptoms on control plants. So that was pretty much a wash. We can say that Xylella fastidiosa was causing that disease because we're seeing the same thing on control plants. Probably due to irrigation, fertilization. I don't know exactly what happened there. Just know that olives don't like to be in controlled environmental conditions. They like to grow outside. You put lights on them, they don't behave very well. Some of the plants were infected by Xylella fastidiosa. I had one plant that was infected for over 2 years. So you just go back and you sample that plant and it always comes back positive for Xylella, no symptoms at all. And never isolated from these plants that showed symptoms. So the question here for this experiment is, well, are the strains of Xylella fastidiosa that we're getting out of olives, can they cause disease in grapevines and almonds? And that was also a control for the previous experiment, inoculation of Xylella back into olives. So someone may say, well, if you take the inoculum, you take the bacterium out of the plant, you grow them in artificial media, and then you put it back in the plant, you cannot expect that to behave the same way as a natural transmission by an insect. So what we did, here's 4 of the isolates that we got from olives. They were inoculated into grapevines, and we did not have Pierce disease on those plants. They were not even positive for Xylella fastidiosa after 3 months, which means the bacterium just died in the plant. When we put those in almonds, we do get almond leaf scorch disease. So that confirms the sequencing analysis showing that it belongs to the multiplex group and not any of the other groups. These are all the strains of Xylella that we have in the lab. We have a collection of strains. So these are Temecula, Stag's Leaf, and M23 that we knew would cause Pierce disease in grapevines. We put them in grapes, and then sure enough, they cause Pierce disease. Also, as a control, as a reference, we used the Dixon and M12 that we knew would cause almond leaf scorch disease. We put them in almonds, and then they cause almond leaf scorch disease. So here, since we got almond leaf scorch disease, that shows that our inoculum was viable. I was not inoculating dead cells into those plants. So that's if the olive strains can cause disease on the grapes and almonds. Now, the other way around is like, are the grape strains and the almond strains capable of causing disease in olives? Olive strains capable of causing disease in olives. For this experiment, instead of using the needle inoculation, I actually used the insects, glycerine sharpshooters. So they were allowed to feed on these plants here, almonds, grapevines, and olive, with all these different strains of the bacteria. So they were allowed to acquire bacterium for 96 hours. Then they were transferred to healthy olive plants in groups of 10. And I think for this experiment, we used Arbequina. And these are the different subspecies. And we did get transmission of multiplex, Xylella multiplex, to 3 out of 26 of these olive plants. We also got transmission of the Rolling Hills, which is an olive strain that was placed in almonds, into olives. So glycerine sharpshooter transmitting the bacterium from one plant to another. The one that scared me the most was this one here. It's a fastidiosa strain that can cause Pierce disease, and that actually went into olives, which means that strains that cause Pierce disease can also use olives as a host. And none of these plants showed symptoms of olive leaf scorch or dieback. They're perfectly fine until we finished the experiment at 12 or 2 years. So this is just kind of a summary of the surveys that we did. Again, we surveyed mostly the San Joaquin Valley and Southern California. No samples positive here. Fresno, Tulare, and Kern County. 3 samples that were positive in Dual-Core. And it's right here in an area that was previously highly infested by glycerine sharpshooters before the air-wide program kicked in. They're not there anymore. Well, they are, but the numbers are very low. And then when we go to Southern California, where sharpshooters are everywhere, then you can assume that these insects are transmitting the bacteria to plants. So that's why most of the samples here were positive. The inoculation rate is so high in Southern California. You have so many insects visiting those plants many times during the day, feeding for hours, that eventually if you pick a leaf from that plant, you will detect Xylella fastidiosa. So the conclusions, if you're going to get something out of this today, I want you to take this here, that Xylella fastidiosa did not cause disease in olives in our conditions. The conditions that we described here. So these are California strains of Xylella fastidiosa. The strains that we recovered from field olives, they behaved as expected for the multiplex group, which is causing disease in olives but not causing Pierce disease in grapes. I haven't seen the fastidiosa group in the field yet. But again, I haven't sampled half of the state. Glycerine sharpshooter transmitted the fastidiosa group to olives, which means that olives can host that one strain that causes Pierce disease. Glycerine sharpshooter use olive trees for reproduction and also as an overwintering host. All the air-wide program, this is the air-wide insecticide application program coordinated by the CDFA. It's targeting glasswing sharpshooters in citrus. Now, olives, if sharpshooters are actually using olives as a overwintering host, may have to revisit that and perhaps have to treat them in the olives as well. We don't have data to support that at this moment. And then, unlike citrus, citrus is not a host for Xylella fastidiosa in California. It is a host for Xylella in South America. Actually, there's a disease, citrus variegated chlorose disease, caused by Xylella in South America, but not here. You cannot find Xylella, not even asymptomatic infections. Apparently, you won't survive in citrus. And then I'd like to quickly comment on the new disease and also a new finding of Xylella fastidiosa in Italy. Something that happened it started in 2010 when they started seeing symptoms of a disease which they call complexo del dissecamento rápido del olive. Olive rapid decline, complex in English. And that's the eye of the hurricane here near Gallipoli on the heel of the boot. And that's something that apparently spread really fast. From 2010 to 2013, it covered an area of about 8,000 hectares. I think right now is about 20,000 hectares. They're affected by the disease. So this is a picture that was taken by a journalist, a local journalist in 2013. You can see all the dieback on the trees. This is a picture that, on the same site that I took in 2014. Then they tried to do some pruning here, trying to eradicate whatever it's causing that. Apparently it didn't work. The suckers are pushing out, but according to the local scientists, you will see symptoms of dieback and leaf scorch on the suckers as well. So trees may not recover from that. These are the scorching symptoms, or the leaf scorching symptoms they're reporting. A little bit different than the ones that we see here in California. They lack the yellowing transition between the brown and the green. They don't have that the V-shaped scorching very well defined, as we see here. Dieback is dieback. A lot of pruning going on, trying to get these trees to recover. Apparently, pruning doesn't work. So the thing, this disease raised the attention of growers in Italy. So the body research team, that's the team that was put together to investigate these new disease. It's led by Maria Saponari and Donato Ossia. So they went out and conducted some surveys trying to find a pathogen or something that's causing this disease. They found 5 different species of fungi. They found Xylella fastidiosa. When they saw that, I think they said, ouch, or something similar to that, you know why. Then they found insects. It's called a leopard moth. It's present here in the U.S. I think it's in California. The numbers are very low. And what the larva does, it just drills into the wood, and that serves as an entry point for fungi, mainly saprophytes. So you can see here the galleries made by the larvae, and then eventually you get the fungi going in and just colonizing the plant. Apparently, the disease is beyond the area where this insect is. So they're sort of discarding this insect as the causal agent of the disease, because you do see the disease where this insect is not present. And then Xylella fastidiosa, initially they saw some cells in the xylem. That was later confirmed by a PCR during the sequence analysis. And as I mentioned, they continued and they pursued some more investigations, and they found that it's this is a strain that they have. It's more related to the strain from Central America and South America than what we have here in California. Not exactly the same as the other strains in that subspecies, but they were close enough that justified shoveling this thing into the Alca subspecies group. It may be a different subspecies group until further analysis. It's a Palca group. So the problems, I was contacted by a number of people back in 2013, and that's what I said in 2013, and that's what I say now. You have two problems. You have a disease, and then you have Xylella fastidiosa. Regardless of the relationship of the two, if Xylella is actually causing disease, in olives or not, they're gonna have to deal with both. The disease, because it's affecting growers, they're losing yield, they're losing trees, they're losing complete orchards. And Xylella fastidiosa, because it's a quarantine pathogen in Europe. That means that all the European Union countries, all the member states are required to eradicate if they find a bacteria. So that's when I mentioned that when they found fastidiosa, they might have said, ouch, we got a problem here. And that's what's happening in Italy right now. So it's mandated to eradicate plants that are infected by the bacterium, regardless if the tree is showing symptoms of olive leaf scorch, dieback, or whatever. If the plant tests positive for Xylella, it must be eradicated. And that's causing some problems with growers in the public. I just read an article in Scientific American maybe 2 months ago, 3. Perhaps that a special armed force police had to escort the crews that are cutting these trees. That's regular police right there. When they say special armed, I guess a shield or something else. So people are refusing to cut the trees. They're finding Xylella fastidiosa in almonds, although it's not confirmed that it actually causes almond leaf scorch disease. Again, well, of course, they're finding in olives, they're finding Xylella fastidiosa in oleander, they're not finding in grapes. Apparently, the strains that they have, they were able to culture, inoculate into grapes, and it does not grow in grapes. Apparently, it grows for a while, for a few weeks, but then eventually the infections die off, disappear. Xylella fastidiosa was also found in southern France, infecting ornamental plants in the genus Polygola. It's a different subspecies group than the one found in than this one here found in Italy. It's in the multiplex group, according to the genotyping. Although the strain that they have in Italy belong to subspecies Pauca, which causes disease in citrus in South America, in Italy it's not affecting citrus at all. As a matter of fact, it's not even infecting citrus. They have tried inoculating trees in the same with grapes. They will grow for a while, but then eventually they die off. Infections die off. So it seems to be concentrated in the olives. And they're also finding some weeds, volunteer plants that grow in the orchards. If you wanna know more about the infection or how widespread this infection is of Xylella fastidiosa, in southern Italy, you can go to this website here, and they do update every 2 weeks or 3 weeks. So I got these December 28th. So the red dots show trees that are positive for Xylella fastidiosa. All the green ones are trees that were negative for Xylella fastidiosa. The sample, cut the map here, but it goes all the way to about right here. But I bet that people are testing plants everywhere as well. Need to be there. There they show the raw data. Actually, they show the reports from the field inspectors where the samples were collected and the lab results. It's not something that was digested by scientists. You just get the whole raw thing in there. You can pinpoint the locations there. As I mentioned, I was there in 2014 in July, and then people are trying pruning techniques. Pruning works a little bit in citrus for Xylella fastidiosa. If you see the symptoms early enough, you can cut that one limb out so you prevent the bacteria from infecting the plant systemically. Number of branches cut off with some brown necrotic tissue. They're trying different weed management programs, trying to reduce the bacterial population. These insects, apparently they like grass a lot, and then they move up to the trees, and then they start moving the bacterium around. They're trying different methods to control the vector. These are some of the historic old trees that they have. Some of these trees can be up to 2,000 years old. Some of these are being affected. They're trying, I think they might try some tetracycline applications or some other treatment just to save those trees. I saw a number of cicadas. They seem to be very common in the area. And the Italians like to listen to these cicadas sing. It's actually a nice sound. In California, we have one species that can actually transmit Xylella fastidiosa. It's not very common. We have the other sharpshooters. They're more common, much more effective, much more efficient in transmitting the bacteria. We have one species, like a desert species in Southern California, that can transmit. I mentioned to the Italians that this could be involved in transmitting bacteria around, but they don't want to touch that. They like the and a number of other insects just feed on the olives, especially when during the dry season, summer season, there's no weeds or anything for them to feed on, so they'll go to those trees. I did see the galleries for the leopard moth, Zeta, and then some scorching, all different kinds of scorching there. And of course, dieback. Trees are dying. The orchards being removed. So if you have any questions, I'm not going to stick around for the reception. I'm going to drive back to Parlier today, so you might want to manage and ask me questions now. And with that, I'd like to thank Chris again for the